Very good morning, dear students. In this class, we are going to discuss the next class of compound lipid, that is glycolipids. The previous class, we have discussed phospholipid, the major types of compound lipid. We have discussed the relevance, the structure, and the different types of compound lipids and their associated clinical complications and the biochemistry relevance. And the next topic illustrated in your syllabus, that is glycolipids. So as you all know, glycolipids is the another important type of compound lipids. So it is a molecular conjugate. As the name indicate, this consists of a glyco, that is a carbohydrate part, and lipids, that is fatty acid part, and that is commonly present in the biomembranes. So along with the phospholipid, these glycolipids are also present. Okay, this is the lipid residue and this is the carbohydrate or sugar residue. Such kind of molecules are known as glycolipids. And the glycolipids are otherwise known as cerebrosides or glycospongosides. Okay, so these are one of the major constituents of brain, where they account for 8% of the solid matter, and this is also present in the various tissues. And there are two important types of glycolipids. One is cerebrosides, next one is gangliosides. And the head group of this glycolipid consists of one or more sugar. Okay, one or more sugar like this, and cerebrosides thus are often known as glycospongosides. Okay, so these are known as glycospongosides because they are exactly like a phosphosphingoside, but there are little bit difference and there is a carbohydrate residues are present. And the glycolipids composed of a hydrophobic region which contains two long hydrocarbon tail, that is fatty acid, and a polar region which contain one or more sugar residue. But the most important thing, there is no phosphate group attached. Okay, and as you can see, this is the biomembrane, the fluid mosaic model of plasma membrane. Here you can see the self-sealing. One of the most important property of fluid mosaic model is they are self-sealing, and the self-sealing properties are mainly due to both phospholipid and glycolipid. Here you can see the hydrophilic head of phospholipids projected towards outside, and the hydrophobic tail faces each other. Okay, there are, that is phospholipids, and in between phospholipids this glycolipid, here I have highlighted in green color. Okay, so this is the glycolipids, where it is embedded along with this phospholipids. Okay, and both glycolipids and phospholipids form the self-sealing lipid bilayer that are the basis of all the cellular membranes. Okay, that is an important aspects. So that is the function of this glycolipids in biomembrane. And if you look the glycolipids, it has lot of biochemistry aspects. So if the glycolipids are, especially the glycolipids, especially the cerebrosides, if they are appear in relatively large amount, and that will lead to a condition called as Gaucher disease. Okay, so the cerebrosides, if it is accumulated in large quantity either in liver or in spleen, it leads to Gaucher diseases. Okay, and this Gaucher disease is otherwise known as glucocerebrosidosis. Okay, and this is an autosomal recessive and hereditary syndrome. And the major reason for this particular infection, generally the cerebrosides are in an adequate quantity, so it will be automatically controlled by the release of the enzyme called glucosyl ceramide hydrolase or glucocerebrosidase. Okay, so this glucocerebrosidase, what will happen, that will cleave the glycosyl ceramide, okay, that is commonly present in the glycolipids or glycospingolipids. Okay, so that is attached in the glycospingolipid, and the bond between this compound will be degraded by this particular enzymes. Okay, and that will leads to glucose and ceramide. Okay, but in the absence of this enzyme, this cleavage will not happen. Then what will happen, the glycospingolipids or spingosides will go and accumulate in the tissues, okay, the cells and the organs like liver and spleen, and it will cause a disease called Gaucher disease. This is an over accumulation of this glycospingolipids. Okay, so this ceramide, generally this glycosyl ceramide hydrolase or glucocerebrosidase, which cleaves the ceramide from the ceramide trihexoside, this compound, which is causing the deposition of this particular component. In the absence of this cleavage will not happen, so that will leads to the accumulation of glycolipids, that is known as Gaucher disease. Then the structure of glycolipids, first one, cerebrosides. Okay, cerebrosides are an important component. Their deficiency over accumulation leads to Gaucher disease, and the structure is almost similar to that of phosphospingosides, okay, or spingomyelins, okay, or that is very important component in the biomembranes as well as the in the biochemistry aspects. So they contain high molecular weight fatty acid called sphingosine. Okay, and either galactose or glucose will be attached instead of the nitrogenous base choline, and the nitrogenous base choline is absent. In the absence of that, galactose or glucose will be present, and in addition to that, there will not be any phosphoric acid. Okay, thus they are not having any electrical charge. If phosphoric acid is there, like phospholipids, they will be able to contribute negative charge, and but here there is no phospholipids, thus there is no electrical charge, and since they are in their polar groups are neutral. Okay, in general properties they exactly same to the spingomyelins, and the major components of this cerebroside is a ceramide. Okay, that is the sphingosine fatty acid. Okay, the sphingosine fatty acid is known as ceramide, that is N-acetyl sphingosine. Okay, N-acetyl sphingosine, that is the cerebroside, and which consisting of the sphingosine fatty acid portion of cerebroside is a ceramide. Okay, and this is the structure. As I said, here you can see the everything is same like a spingomyelin. So the structure is CH3 CH2 12 times CH, there is a double bond CH and CHOH, there is an alcohol here, and CH NHCO, the fatty acid side chain here. Okay, and CH2OH, and that hydroxyl group will be attached with the galactose. Okay, instead of choline. Okay, so that is a structure here. Hydroxyl, the hydroxylation. Okay, the hydration. Okay, removal of water will take place here. Okay, and here this is the structure here. So the galactose moiety, sphingosine fatty acid, and there is no phosphate residues. Okay, that is what I told you. The sphingosine carries the galactose by glycosidic linkage or its primary alcoholic group. Okay, and the fatty acid chain, okay, in this amide linkages of primary amino group. Here the fatty acid is present in the amide linkage, and the galactose is present in the primary alcohol linkage. Okay, so this is the structure of cerebrocytes. And the cerebrocytes are classified into various group depending upon the fatty acid composition. Okay, so depending upon which type of fatty acid is present, that can be classified into 4 different groups. One is kerosene, next one is phrenosine or cerebron, next one is nervine, next one is oxynervine. So these are the major types of cerebrocytes. Okay, now we will see very briefly about all those components. In the case of kerosene, I told you the major difference is the fatty acid. Okay, so what type of fatty acid here it is present? In the case of kerosene, everything is the same, but here the fatty acid, this R will be, what is that? That is C24 lignoceric acid. Okay, C24 lignoceric acid, that is specialty of kerosene. Then the phrenosine or cerebron, so it contains 2-hydroxy derivative of lignoceric acid called a cerebronic acid. Thus it is known as a cerebron. Okay, so that is again very, very important. Then in the case of nervine, it contains an unsaturated homologue of lignoceric acid called a nervonic acid. Okay, so here it consisting of an unsaturated homologue of lignoceric acid called a nervonic acid. Thus it is known as a nervine. Next one is oxynervine, which consisting of another 2-hydroxy derivative of nervonic acid. The hydroxy derivative of this component called a oxynervonic acid. So these are the major different types of cerebrocytes. So here these cerebrocytes are mainly 4 types, and here it is. One is lignoceric acid. This is a structure of lignoceric acid. I told is a C24, here 22 and 2. Okay, total C24 lignoceric acid, CH3, CH2, 22 times COOH. That is the structure of lignoceric acid. So that is mainly present in kerosene. Next one is, here it is cerebronic acid, CH3, CH2, 21 times CHOH, COOH. Okay, that is commonly present in phrenosine or cerebron. Okay, thus it is known as the presence of cerebronic acid is known as that is why this compound is known as cerebron. Then next one is nervonic acid. Here the structure is CH3, CH2 7 times. There is a double bond. I told you there is an unsaturation. Okay, CH2, CH double bond, CH, CH2 13 times and COOH, nervonic acid. And similarly here also an unsaturated acid, that is the hydroxy derivative of nervonic acid called a oxynervonic acid, that is CH3, CH2, CH, CH2, CH2 12 times. There is CH instead of this one CH2, one H is replaced as hydroxyl group, CHOH, then COOH. So these are the different types of cerebrocytes. Then the second group of lipids are known as gangliosides. So in the year 1955, a great scientist called Planck, okay, he found out a special type of glycolipids from the brain tissue, and it is commonly present in the ganglionic cell of nerve tissue. Okay, so thus it is known as a ganglioside. Okay, the lipid that is present in the ganglion cell of the nervous tissue, that is known as a ganglioside. And this is another another tissue such as parenchymatous tissue like spleen and erythrocytes. Also these gangliosides are very commonly present, and this is constitute approximately 6% of the membrane lipids in the gray matter of the brain. Okay, but it is found very less quantity in the biomembranes of most of the non-neural tissues. Okay, and the gangliosides act as the receptors for the toxic substances like the pathogen, especially the bacterial pathogens such as Vibrio cholerae. Okay, that is a causative organism of cholera. Then the influenza virus. Okay, and tetanus toxin. Tetanus, as you all know, that is caused by Clostridium tetani. Okay, and this gangliosides plays a very vital role in the cell-to-cell communication or cell-to-cell interactions. Then the structure of ganglioside, the structure is little bit complex. Okay, structure is little bit complex. The structure of gangliosides is a complex and related cerebrosides in which contain a ceramide. Okay, that ceramide is N-acetyl sphingosine, which are commonly connected or linked to a carbohydrate, either glucose or a galactose. That is what I told you. A common carbohydrate sugar moieties are present. That is why it is known as a gangliosides. Okay, and generally it contains 2 additional mole of carbohydrate, that is either glucose or galactose, and 1 mole of N-acetyl galactosamine, and 1 to 5 mole of N-acetyl nuraminic acid or NANA. Okay, so these are the structure. So these are the ceramic residues. Okay, so it has long chain fatty acid residues, and here you can see GLC stands for D-glucose, and GL stands for galactose, the NANA stands for N-acetyl nuraminic acid, then GalNAc stands for N-acetyl D-galactosamine. Okay, and there you can see that's another galactose. Okay, here 2 galactose and 1 glucose are present. So like that you will be having 1 to 2 additional moles of carbohydrate. So that is another important aspect. Glucose, galactose, acetyl nuraminic acid, N-acetyl D-glucosamines are the major component of gangliosides. Then this is the structure. This is a very complicated structure, a detailed structure here. So this is CH3, CH2 12 times CH, 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 OH, and CH2, CH, CO, CH2. This is the sphingosine part. Okay, the fatty acid part, and which connect a D-glucose and also connect a D-galactose, and that connect with the N-acetyl D-galactosamine. Okay, and this again, this galactose residue is connected with this particular residue, that is known as a spingolipid GM2. Okay, this is known as a Tay-Sachs gangliosides. Okay, so this is the structure of a ganglioside. Then like glycolipids or cerebrosides, in the case of ganglioside also another important biochemical implications are there. Okay, so that biochemical implications are known as a Tay-Sachs disease or GM2 gangliosidosis. Okay, GM2 gangliosidosis. So these gangliosidases are storing large amount in brain and spleen. Okay, the GM2 gangliosides are stored in large amount of brain and spleen, that leads to GM2 gangliosidosis or Tay-Sachs disease. Okay, this is first identified by an English physician called Warren Tay and an American neurologist called Bernard Shaw. Okay, so that is known as Tay-Sachs disease. Okay, Bernard Shaw and Warren Tay identified this infection. So this disease. Okay, and this disease is mainly caused due to the deficiency of enzyme N-acetyl galactosaminidases or hexosaminidase A. Okay, so what will happen is this hexosaminidase, in general case, if sufficient amount of gangliosides are present, this saminosidase enzymes will be produced, that over accumulated gangliosides will be cleaved in this particular region. Okay, N-acetyl D-glucosamine and D-galactose residues. Okay, and this will cleave the specific bond between beta-8,4 linkage between N-acetyl D-galactosamine and D-galactose residues, the polar head of the ganglioside GM2. Okay, so this is the polar head of the ganglioside GM2. It will be making the cleavage, and what will happen? And it will be cleaved. But in the absence of this enzyme, this cleavage will not happen. These gangliosides will go and accumulate a large amount in brain and spleen, that leads to Tay-Sachs disease. And the Tay-Sachs, this particular glycolipid GM2 are known as Tay-Sachs gangliosides. gangliosides. Okay, and this Tay-Sachs ganglioside is accumulated in the large amount in the lysozyme of the brain, approximately 300 to 300 times more than the usual content of GM2, which cause the degeneration of the nervous system. Okay, so the Tay-Sachs gangliosides accumulate in the large amount in the lysosomes in the brain cells and cause the degeneration of the nervous system. Okay, and this condition is known as gangliosides. So this is all about the cerebrosides, dear students. Okay, so once again with this we can conclude. Once again we can glance through the major points studied in this class. We have discussed what are glycolipids, another important types of compound lipids. They are classified into two groups, cerebrosides and gangliosides. What are cerebrosides and what are gangliosides we have discussed. And the glycolipids, their importance in the biomembrane. Okay, the self-sealing capacity of biomembranes are due to this glycolipids. And the glycolipids, the clinical implication called Gaucher's disease or Gaucher syndrome, and the structure of glycolipid we have discussed. Cerebrosides, okay, we have discussed. Then the classification of cerebrosides. Okay, then we are having 4 different types of cerebrosides: oxynervin, nervin, phrenosine and kerosene. Okay, then the gangliosides are another important component, and here we have discussed the structure of gangliosides, and here the gangliosides, the clinical implication we have discussed, that is Tay-Sachs disease. So these are the major concept we have discussed in this particular class. So as I said, Principles of Biochemistry by Leninger is the major textbook, along with the Biochemistry by J.L. Jain you can refer, and all this textbook you can refer. These are some of the reference book as well. Okay, so thank you dear students for watching this particular class. If you are having any clarification, you can ask me.